Hi, I am Umar Khan Pathan. My topic is DNA is a carrier of genetic information, and the concept is that is Griffith experiment. For a long time, it is believed that the protein of the chromosome were responsible for the transmission of hereditary character from one generation to the next generation. But recent development in molecular biology revealed that the nucleic acid of the chromosome are responsible for the transmission of hereditary character. The nucleic acid which carries the genetic information is DNA. It is illustrated by a number of experiments using bacteria and viruses. So one of the experiment, that is Griffith experiment. Frederick Griffith experiment was in 1928. This experiment demonstrated how bacteria can transfer genetic information. This experiment suggested that the bacteria can transform into a different strain, and such a process is called as transformation. Griffith given a principle of transformation. What is transformation? In which a process in which by DNA from outside a bacterial cell is introduced into the cell. Now we will see on the basis of this diagram how this dead bacterial DNA is transformed into the live bacteria, and these live bacteria become virulent or pathogenic. If this live bacteria is non-pathogenic, it means the character of pathogenic or virulent bacteria is transferred into live bacteria. That's why this non-virulent bacteria become a pathogenic. Finally, it confirmed that the dead bacterial DNA is transformed into a live bacteria. With transformation, the character of dead bacteria is also transformed into the live bacteria. That's why this live bacteria become a pathogenic. And such experiment given by Griffith, it is proved that the character or DNA is transformed from one generation to next generation. So what was the experiment? Griffith studied two strains of Streptococcus pneumoniae bacteria. That bacteria causes a pneumonia in mice. So he selected one strain, that is virulent strain. Other strain is non-lethal. And Griffith injected mice with the heat-killed virulent strain, the non-lethal strain, and a mixture of two, that is both non-virulent and virulent strain to the mice. The mice infected with the mixture died, but the mice infected with the either strain alone survived. So what did the experiment show? It means the non-lethal strain had been transferred into the virulent strain by an unknown heritable substances from the dead strain. These substances become known as the transforming principle. What is the significance? The Griffith experiment paved the way for many advances in medicine and for the discovery that the DNA is the genetic material. So now we can see the experiment that did by Griffith. In first diagram, we can see the rough strain or non-virulent strain is given to the mice. The result is mouse is live. In second strain, smooth strain, that is virulent strain, that is given to the mouse. Mouse is dies because this is smooth strain is virulent strain. In third diagram, we can see the rough strain and heat kill smooth strain. Both are given into the mice. The mice will die. And fourth diagram we can see the heat kill smooth strain is given to the mouse, but mouse is live. So on the basis of this experiment, it is concluded that this rough strain, is non-virulent strain. This rough strain is become virulent because of smooth strain. Heat kill is smooth strain. It means heat kill is smooth strain bacterial DNA is transformed into rough strain. Because of that, this rough strain become virulent. So finally, the Griffith experiment proved that the way for many advances in medicine and for the discovery that DNA is a genetic material. Thank you.